Let's talk about the recent publication that showed that COVID-19 mRNA vaccines sensitize certain cancer patients towards the specific therapy called checkpoint immunotherapy, checkpoint inhibitors immunotherapy. Now everyone jumped to conclusion, hey, that's so great, COVID-19 vaccines can help treat cancers. Well, couple stipulations. This is a very specific type of treatment, so it's not available for everyone. It often doesn't work. When it works, for the select few, it's fantastic. And it's also the same therapy that has led to the discovery of turbo cancers, and I'll talk about that in a second as well. And another key point here is that in order for this to be effective, this therapy had to be used within 100 days of vaccination. And if you went outside of that short time window, then all bets are off. And in fact, the data from the same article suggests that not only did this protection or did this sensitivity wane, it also suggested that non-vaccinated might have been even better off than vaccinated if the vaccination was more than 100 days away from use of this immune checkpoint inhibitors therapy. My name is Dr. Mikola Rashek of Maragenomics, and let's break it all down. Clearly, this will not be a short video. This is why I gave you a summary right now of what was found. And I do need to give you a bit of a background. Okay, so what's going on with cancers? What happens is they stimulate specific reaction, but immunological reaction. So they produce signatures, molecular signatures that are foreign or different from our body, and there are these receptors that can recognize all these foreign signals. These receptors can recognize invaders like pathogens, but they can also recognize cancer. So, and they are called pattern recognition receptors, and they're everywhere. And these receptors recognize everything under the sun in order to recognize an issue. Once they recognize an issue, it leads to release of this type of molecules called interferons, specifically type 1 interferons. Let's not get into crazy details here. What these interferons then do, they themselves promote activation of antigen presenting cells. Now, what are those? These are the type of immune cells that can then start taking a problem, such as pathogen or cancer cells, destroy it, and then take fragments of it and present it to the immune system. They present it on these receptors called major histocompatibility complexes. We have two types of these, type 1 and type 2. And the only difference is what type of immune cells they present fragments of these issues to the immune system. And type 1, for example, presents it to killer T cells, which is the key to our story today. So let's focus on that. Type 2 presents it to helper T cells, and that would help to elevate the reaction. But killer T cells will eventually be the ones, once they are presented with a fragment of what they're supposed to recognize, then they will go and destroy it. And that's these killer T cells, also called CD8 T cells. And the issue here is that then not all cancers can these T cells go in. Why is this? And do their job and kill the cancers. And why is this? It's because of the following. Remember I mentioned those interferons activating these antigen presenting cells? Well, cancer cells can also recognize, hey, interferons are being released in the environment, and as a consequence, they will start producing a molecule called PD-L1. So, and it's because cancer cells can use that information to start tricking the immune system to make sure, that immune system is no longer properly recognizing it. Why is this? It's because when killer T cells recognize what they're supposed to be destroying, what they do is they start producing this molecule called PD-1. That stands for programmed cell death 1. And these molecules, when they first show up, it shows you that T cells are being activated. And it's a sign that, hey, everything is going well. Eventually, if that molecule keeps persisting and there's more of it on these T cells, it means those T cells are exhausted and they will eventually stop working. So it, depending on when you see that molecule PD-1, originally at first it's a signature that T cells are active and doing their job, and eventually they can be overworked. And that's when you have too much of it and it results in T cell exhaustion. Now, cancers can take advantage of this and produce a molecule, PD-L1, that will interact with the PD-1 on T cells, and that's how you shut them off. Why do T cells even do it? It's like a feedback loop so that they don't overactivate themselves and they don't start destroying tissues. So they start producing these killer T cells, starts producing these molecules so that eventually those antigen presenting cells also recognize that there's too much work going on because killer T cells are producing their own specific cytokines, which are molecules that are used by immune system cells to communicate amongst themselves. And antigen presenting cells are recognizing these cytokines produced by killer T cells, and they start producing PD-L1. L, by the way, stands for ligand, and ligand is a generic word for anything that interacts with the receptor. So PD-L1 is a molecule that will bind to a receptor, which is PD-1. And this is how you end up start shutting off the entire system so that these killer T cells just don't go overboard and overkill. But cancer cells can do it as well. Now, this is finally when the immune checkpoint inhibitors come in. And the reason why is because those are antibodies that will target PD-1 on T cells. Why? Because when you put these antibodies into the tumor or cancer environment, and these antibodies now interact with PD-1 on killer T cells, cancer can no longer stop killer T cells from doing their job and therefore killer T cells will do their job and kill the cancer. So this is why this is an effective therapy. Now, in order for this to be effective though, you have to have tumor environment that has shown a lot of activity already. So this is what is referred to as tumor has to be hot. So you have to see the presence of this interferon in that environment. You have to see presence of PD-L1 on cancer cells, because it means these cancer cells, if you see a lot of PD-L1, they were being targeted by killer T cells. You have to see T cells in that environment with that PD-1 molecule, and that's all indicative that this tumor is hot, and such tumors will be a good target with this therapy. Alternatively, you can have cold tumor, and cold tumor is the exact opposite. There's not much of a signature that immunologically these tumors were targeted, so there isn't much interferon in there. There is no many infiltrating killer T cells in that environment, and tumors are showing low levels of PD-L1 molecule. So if you inspect tumors and they have low PD-L1, they are not good targets for this immune checkpoint inhibitors therapy. And if they have high PD-L1, those would be good targets. So that's a bit of a background in order for you to understand what is going on. And then one more thing I'll mention is that, well, when these immune checkpoint inhibitors came on the scene, we started seeing these abnormal type of cancers upon treatment with these immune checkpoint inhibitors, where the cancers, instead of becoming hot and and being successfully treated, they became super hot and in a bad way, where cancers instead became super aggressively growing fast. And in essence, we're talking about turbo cancer. So turbo cancer is a very real thing. I know a lot of people online say there is no such thing as turbo cancer. It is. It's just called something else. We call that hyper progressive disease, and turbo cancer is just a colloquial street name, if you will. But it is really exists, and it is through these medications that we're talking about today that turbo cancers have ever been discovered and studied. And check out my videos on turbo cancers just to see what are the relationship between COVID-19 mRNA vaccines and what we are also observing in turbo cancers as well. So nothing is simple in biology. All right, so that is the background, and this is what these guys, the scientists were showing, that it's the COVID-19 vaccines that sensitized certain types of cancer to that type of inhibition, that anti-PD-1 antibodies. Okay, so what did they check? They looked, this is the MD Anderson Clinic in Texas, and this is what they were looking at. They looked at patients who were either vaccinated versus non-vaccinated. This was between 2015 and 2022. And they looked at just over 700 non-vaccinated versus around 180 people who were vaccinated. About half of them, 80 some, were vaccinated with one shot, 90 some were vaccinated with two mRNA shots. And let's just go from there. And what they were able to show is that those who took the shot and received that immune checkpoint inhibitor therapy within 100 days, and again, this is key, they showed better survival than those who did not. And they investigated two types of cancer. They investigated non-small cell lung cancer. So I'm just going to call it lung cancer from now on. And they clearly showed this survival curve, and it shows that those who had the vaccine within 100 days of using that therapy, their survival rate was almost twice as good as than those who never were vaccinated. And you can see those who had the vaccine, their survival rate was just over 50% by the end of the study, which was in about 3 years, versus those who were never vaccinated, their survival rate was about 30%. So those are the differences. Now, underneath that graph, you also see these numbers, and this is a little bit tricky because you can see that by the end of the study, there was only 3 vaccinated people, as opposed to higher number for the non-vaccinated people. So how can you have 50% survival when there were only 3 people left in the study by the end of the study from the 180? And the reason why is because it's not just, they don't lose patients who die only, they also lost patients simply from the study. While they were still alive, they lost contact with them or the patients withdrew. And when that happens, you continue the study assuming that such individuals will follow the pattern of survival the same way as anyone else remaining in the study. So what this means is that I suppose that they must have lost few dozen people to follow up, or they withdrew. And if those people drop dead the next day, then that survival curve would obviously be quite different than what we're seeing. Nevertheless, statistically, this is statistically significant. That curve could have been either worse or better, but most likely it would still be better than non-vaccinated. So keep that in mind. And they've done many different of these types of analysis, and they continue to show the same pattern, including when the loss of patient, that attrition of patient loss was similar between vaccinated and non-vaccinated. The survival was always better for the vaccinated, again, provided that you took vaccine within 100 days of initiating that therapy. All right. So then what's also interesting here is that this effect, the sensitization to this treatment was only observed for that treatment. If you took the COVID-19 mRNA vaccine versus not, there was no difference for chemotherapy at all between those two groups. They did investigate that. And also other types of vaccines did not have an effect. So influenza vaccines, no impact on sensitizing towards that immune checkpoint inhibitors. Nope, nothing. And other types of vaccines. So specifically, it was mRNA vaccines. And then they also checked melanoma. Melanoma cancer patients that had fewer, they're just over 200, but the pattern is still the same. Those who took the mRNA shots within 100 days had better outcomes when they were treated with this rare treatment option. All right, then they wanted to investigate as to how this really worked, what's going on. And they decided to look at mouse studies because this allowed them to investigate great details. And believe it or not, they recreated their own Pfizer vaccine. Pfizer vaccine information is publicly available, so they were able to literally recreate the RNA, they rebuild the RNA, they rebuild the lipid nanoparticle, and they remade the vaccine from scratch on their own, so they didn't have to depend on Pfizer or Moderna. And they treated mice, specific type of mice that showed either melanoma or lung cancer tumors, and they showed that when they were treated with both this remade vaccine as well as those immune checkpoint inhibitors, tumors decreased. So there's that. But they also showed that this was dependent on that interferon, because if they attacked the interferon or basically removed it, if they were targeting interferon, it might have been with antibodies, then this completely stopped, this benefit stopped. And if you provided the vaccine and injected the mice with interferon, then again there was improvement in terms of reduction of, say, the tumors. So clearly interferon is the key here. They also showed that once the vaccine and the treatment was provided, it elevated activation of those antigen-presenting cells. So showing everything that we know how tumors in ideal scenario should be targeted and treated by the immune system. And it also correlated with increased activation of T cells that could target these tumors. And it also correlated with these tumors showing higher levels of that PD-L1 molecule as well. So everything fits. But what was also cool is that this allowed them to study further as to what aspect of vaccine was it that resulted in this. And this is the really cool part. It didn't have to be spike protein. They changed the vaccine and it coded for a completely different virus protein, and the effect was the same. So it had nothing to do with the spike protein. It has to do with the construct of the vaccine, and specifically it was the genetic material within the vaccine that caused this. And what they suggest is that the genetic material can, even though in the COVID-19 mRNA vaccines, the genetic material was constructed with these synthetic uridines that are rarely found in nature. It was done in order to avoid recognition by the immune system. What the authors propose is that the genetic material can still create a certain three-dimensional structures that are recognized by those pattern recognition receptors, and the whole thing starts. And that's why the vaccine can sensitize temporarily the immune system to be able, or sensitize cancers to be able to be well attacked by this therapy, by these immune checkpoint inhibitors. What they also did is they also changed those synthetic uridines for normal uridines, and that made it even better. So meaning the immune system was activated even more powerfully, and the treatment of tumors was even better. So that's the breakdown, and they show this very cool figure showing you what they think is happening. So when tumors are not recognized by killer T cells, by providing the vaccine leads to activation of those pattern recognition receptors, which lead to release of these cytokines. Cytokines activate your antigen-presenting cells, which then present information to killer T cells as to what they're supposed to be targeting. Killer T cells can then go to the tumor and attack tumor, but the tumor prevents it with this PD-L1 molecule, and this is finally when the antibodies come in that will stop that interaction between tumor and killer T cells, and then killer T cells can do their job and kill the tumor. So that's the image right there. But they did something else, and this is finally going to bring us to why this time window is so important. So they also wanted to analyze how often did the vaccines sensitize cancers that they had in their data bank, how often was this observed? And specifically, what they wanted to measure is how often, once people who had cancer were vaccine recipients, how often did their tumors have that elevated level of PD-L1 molecule? This is referred to as tumor progression score, tumor progress score, TPS, can't remember what it stands for. And the point is that if the tumor shows less than 1% of that PD-L1 molecule on its surface, that's not going to be receptive to this checkpoint immunotherapy, immune checkpoint inhibitor therapy. It's just not going to work. Anywhere between 1% and 50%, it's in the intermediate range, and anything above 50%, so anything between 50 to 100% of the tumor cells showing presence of that PD-L1, those are candidates to be very effectively well treated by this therapy. And they looked at more than 2,000 samples. I think it was 2,300 some samples that they had historically collected at this institute. About 2,000 of these were non-vaccinated people. And then they had the remainder was vaccinated group, and they divided it into two types of groups. Those who were vaccinated within 100 days of using this immune checkpoint inhibitor therapy, and those who were vaccinated more than 100 days. And they measured these levels of PD-L1 in these tumors. And this is where it finally gets interesting, because what they showed is the following, that those who had vaccination within 100 days of that treatment, they show elevated levels of PD-L1 in comparison to non-vaccinated. And that increase was about 24%. Okay? But here's finally the point that I also want to tell you, that time window is really important, because they also compared the vaccinated within 100 days and those who were vaccinated but more than 100 days from when the therapy was used. And when that happened, the vaccinated within 100 days were showing 41% higher levels. Their tumors of people who took vaccines within 100 days of using that therapy, their tumors were 41% more often showing more PD-L1 than those who had the vaccine more than 100 days. And if you actually compared the vaccinated who were vaccinated more than 100 days away from using this therapy with non-vaccinated, they were actually worse off. And you can see that in this graph that looks like there's these flasks, right? These flasks, what they're actually showing is how many of the people whose biopsies were looked at for this PD-L1 level. It shows you how much percent of their cancer cells show that PD-L1, and you can see the bottom is this fat part of the flask. That's because majority of all the people showed very little percentage of that PD-L1 on their tumor sites. And then we're interested in anything above 50, and you can see the differences between vaccinated within 100 days of using that immune checkpoint inhibitor therapy, or over 100 days and non-vaccinated, and you can see non-vaccinated had more PD-L1 than those who were vaccinated but more than 100 days away from using this checkpoint inhibitor. So there is something going on there where eventually this sensitivity wanes, it disappears, and potentially, although this would require more investigation because this was not statistically significant, potentially this, not only does it wane, but it seems like it might get worse over time. So the nuances are important. Now, long time ago, I did a video warning about this synthetic uridines in the mRNA cancers, that they might desensitize the immunological response, and that's what might lead to the development of cancers. And this was hypothetical. We published a paper with a group of scientists on that as well. And the results of today's paper might seem counterintuitive or contradictory to that, but again, it all comes down to time, because the theory still is applicable provided what time point we're looking at. Because remember, these pseudouridines allow these mRNA constructs to last longer than would be expected for natural mRNA, and as we just seen in the study, they don't activate the immune system to the same degree as if natural, normal mRNAs would. And as a consequence, what might be happening is that such construct could have a low-level chronic stimulation, and eventually that could turn the immune system towards tolerogenic state, meaning it just simply stops ignoring it. And that could result in, for example, elevated levels of PD-1 molecules, whether that's antigen-presenting cells or regulatory T cells, elevated levels of M2 macrophages, which also stop inflammatory immune response. They quiet things down. It can lead to elevated levels of IgG4, and as a consequence, you suppress the immune system. And here we are seeing potentially an example of that as well, where over time, more than 100 days after vaccination, those who are vaccinated, they no longer are showing same level of tumor sensitivity as if people who were never vaccinated. So I thought I would wrap that up, add that in there, that theory we mentioned still could be applicable, but we need more data, collect more data to really find out. All right, told you this is not going to be easy, simple, quick video, but I finally wrapped it up. And last thing I mentioned, please check out my Patreon channel. Why? Because two things. Number one, my supporting patrons get to vote as to what comes out on YouTube, number one. And number two, anyone, whether you're supporting patron or whether you're free patron supporter, anyone can now contribute towards this project I'm working on, which is collecting information from my audience as to how to build a guide towards the greatest, best way to conduct healthy living. And I want information from you to help me do this. This is something that I want to have available for free to anyone. As I keep mentioning, is that post-pandemic, because of the spike protein interaction that we all had to endure one way or another, the best way to protect ourselves is now via healthy living. And I want to find out from you, what does that mean? I know what that it means for myself, but that doesn't mean it has to be correct. And I want to know what it means for everyone else and start building a guide as to what that means. So now I have also free webinars where we can all interact, hang out as well. So live online interactions where I'll collect that information from you, and I'll see you hopefully at the next event. Bye everyone.